Hi everyone, my name is Or Zolti and I'm a PhD candidate in the Nanoelectrochemistry lab at the University of Georgia under the supervision of Dr. Ramaraja Ramasamy. The project I present today is our work on a biosensor for rapid detection of Listeria monocytogenes for food safety applications. Listeria monocytogenes is a foodborne bacterium common in different variety of foods such as deli meats, chicken, eggs, dairy, beef, pork, and produce. It is known to grow in temperatures as low as 0 degrees Celsius, which makes it a danger for ready-to-eat foods, refrigerated and frozen foods. Listeria monocytogenes can infect the brain, spinal cord membranes, and the bloodstream of the host. It is the third most common cause for meningitis in newborns and may cause miscarriages, stillbirth, and preterm labor for pregnant women. The FDA reported that Listeria outbreaks has a hospitalization and mortality rate of 60% and 11% respectively out of confirmed cases, and it cost to the public is around $3 billion annually through to 2014. It is clear why detection should be a priority in the food industry and why a robust and accurate rapid detection method should be developed. Mostly because the current detection standard involves periodic sampling, shipping the sample to a laboratory, culturing the samples, plating them, identifying the bacteria, and then counting the colonies. This long process takes about 24 to 72 hours without shipping times. It requires a highly trained personnel and expensive equipment, which sky high the price per test. The biggest issue is that supplied goods might be recalled because of the time it takes to use the current standard, and that is why we had the interest in creating a very rapid and accurate testing. There are different approaches to how to detect Dysteria, and the use of biorecognition element is a great way to do it due to their natural ability to attach to a single specific target. These elements can be antibodies, protein, DNA or RNA, enzymes, or bacteriophages. From all these methods, phages tend to have the highest environmental stability, and due to its commonality in nature, their cost is lower with respect to the rest of them. Phage also has the ability to distinguish between live and dead bacteria, which gives an extra edge when we look for a biorecognition element. Electrochemical biosensors have very high sensitivity and the ability to provide almost immediate responses. That's why we decided to combine an electrochemical biosensor with the bacteriophages to also add specificity to the already fast and sensitive electrochemical biosensor. The image on the bottom left, you can see how we modify the electrodes by adding carbon nanotubes and modifying them with the phages. The current requirements from the FDA is that food will have less than 10 CFU per gram for vulnerable population consumption and 100 CFU per gram for general public. In this study, we did testing in triplicates and achieved the limit of detection of 8 CFU per ml, which is way below the standard. You can also see the response for different concentration in the image on the top middle, and below it you can see the calibration curve of said sensor. It is very accurate and it is very, very sensitive. After finding the detection limit, we tested how the sensor is working with and without the biorecognition element. You can see on the top left bar chart that without the phage, we get no signal for 10 CFU per ml, and above it, the signal is almost constant. Unlike it, once we added the phage, you can see a linear response to different concentration of phage, which also confirmed the previous tests we did. On the right of that, there is another bar chart that shows you the effect of the biorecognition element on non-target bacteria. So here we tested Listeria, and also we tested Salmonella and E. coli. We can see that without phage, E. coli and Salmonella get higher responses, but after adding the phage, Listeria now the signal rise by 30%, and the Salmonella and E. coli was dropped by almost 20%. In addition, we conducted Instability Study, that's the bottom bar chart, and you can see here the responses after 1 hour, 1 day, 1 week, and 2 weeks after the deposition of the phage on the electrode. It is clear that those electrodes can be used even after 1 week to achieve very stable results, which is a very good indication for the promise of this sensor. In conclusion, we have shown here a sensors with detection element way before the requirements of the FDA and with detection time of almost 10 minutes. This detection time with the stability will allow the integration of sensor like this into existing production line to achieve real-time detection and reduce the use of outside expensive labs. Thank you for listening, and I'm happy to have any questions.